Okay, Nora.
Okay, so we're going to start a unit on protein synthesis. Protein synthesis is a fancy word to say making proteins, and we do that by reading coded instructions from your DNA. I like to think of your DNA as sort of the foreign language instructions to writing how to make proteins. So you have your DNA, which are your foreign language instructions, and every single one of your cell has a complete list of all the instructions, okay? DNA is made of deoxyribose sugar, a phosphate group, and a nitrogen base. Together, those three things make a nucleotide. And we know that my nitrogen bases are adenine, guanine, cytosine, and thymine. A goes to T, C goes to G. All right? DNA shape is a double helix, and its job is to stay in the nucleus where it is protected and safe. When I want to make a protein, I will isolate a gene on my DNA, a small portion of it, and I will make a copy. I will essentially take my foreign language directions and I will translate them into English so that I can read what those directions are telling me. That translated version of my directions for making a protein is called RNA. RNA is a little different than DNA, and I've circled the differences so that you could see. So RNA is still made of a nucleotide. It's got a 5-carbon sugar, although this time it's called ribose, a phosphate group, and a nitrogen base. 3 of the nitrogen bases are the same as DNA, adenine, guanine, and cytosine. But in the place of thymine, you have a nitrogen base called uracil. So if I had a DNA strand that looked like this, and I wanted to make it into mRNA, which is a type of RNA, that's my translated instructions, C still goes to G, but instead of A going to T, A goes to U. And if you see a T, it goes to A. Yes? Yeah.
Why would it go to A?
What?
Why would the T go to A?
Because T always pairs up with A anyways.
Oh.
Okay? So mRNA, almost the same. It's a disposable copy. It is single-stranded, so it doesn't look like a double helix. It actually looks like a comb, like that you would use to brush your hair. So it's got the backbone, and then it's got the bases that stick out the side. And finally, RNA, because it is an English-translated copy of my directions, is able to move out of the nucleus and be read and turned into a protein there. Okay? So go ahead and write this stuff down. Into your notes. Both DNA and RNA are made of long chains of nucleotides made of a 5-carbon sugar, a phosphate group, and a nitrogen base. And they are both considered nucleic acids. However, some differences in that RNA is called ribonucleic acid because the RNA's sugar is ribose instead of deoxyribose. It is single-stranded, not a double helix, and they have the base uracil instead of thymine. We talked about how RNA is nothing more than just a disposable copy of a DNA segment, that segment being the gene that we want to translate into a protein. So we could take one gene in DNA and copy it into many, many, many copies of RNA, as many copies as we want to make proteins. And there are 3 different types of this disposable DNA, or RNA as we call it, that do work to make a protein. The first type is mRNA, or messenger RNA. I remember it because it carries the gene message that we've translated. The ribosomal RNA, just a fancy way to say ribosome, we call it rRNA. And tRNA, or transfer RNA, its job, where mRNA are the directions for how to make the protein, the tRNA is what actually does the building. The building blocks of proteins are called amino acids. So tRNA's job is to figure out which amino acid the mRNA is coding for, and then bring that mRNA, or bring that amino acid to the ribosome to build it protein. So mRNA is the RNA that carries copies copy of the gene. It acts as a messenger. The second type is the rRNA, fancy word for ribosome. And this is where the protein is actually put together. So think of it as the construction room, where we're going to bring the amino acids together. The mRNA actually passes through the ribosome, where it gets read and decoded to say what amino acids we need to make the protein. So the rRNA is the big red one. And the last RNA is the tRNA. This RNA is found only in the cytoplasm, and like I said, where the mRNA is the directions, the tRNA is what does the actual building of the protein. It brings the amino acids to the ribosome and helps build a protein using those amino acids linked together. Homework is 13.2. It is due tomorrow. Okay, so yesterday we talked about, very briefly, how DNA is a code in our bodies that tells us how to make proteins. And I said yesterday that I like to think of the DNA as the foreign language version of our code, and we have to translate it into English so that we can understand it. And that translation is turning it into mRNA. Okay, mRNA stands for messenger RNA. It is a throwaway copy of a gene. We use it simply for the process of reading it to make proteins, and then we get rid of it. So, when you make a protein, transcription is the first process that you do. Transcription is taking a segment of DNA called a gene and copying it into mRNA. And if you remember, there's a couple differences, including that mRNA has the base uracil instead of thymine. So remember when we did DNA replication and you had one side of the strand and I asked you to make the other one, A to T, C to G, and so on? Okay, same thing when you're going to make a piece of mRNA, except if you have an A, it's going to hook up with a U instead of a T. Okay, so here's my picture. This is a snapshot of the nucleus of a cell. And we know that inside of the nucleus there are 46 chromosomes. I drew 45 little ones over here, and my 46th one is a big chromosome, so that we could see that this right here is the gene that I want to eventually make into a protein. Okay, so I only drew one side of my DNA. I didn't do both. I only did the one side because I couldn't really fit it in there, and we only really need one side anyways. All right, so the first thing that happens is I isolate the gene that I want to turn into a piece of mRNA, my throwaway copy. All right, and I begin by laying down mRNA nucleotides. They look just like DNA nucleotides, except they have a U instead of a T. So where there's an A, I would put a U. Where there's a T, I'd put an A, and a C, and a G, and so on and so forth, until I've taken this entire half of DNA and turned it into a piece of mRNA. That's my copy. I've gone from my foreign IKEA directions to my English directions. Now my cell's going to have a really easy time reading that piece of mRNA to make a protein. Right? But we're not done yet. Because sometimes when we're laying down mRNA base pairs, we might add too many base pairs, and our body needs to know that some of those are not going to code for my protein. They're not part of my directions. It would be like baking a cake, and somewhere in the directions it says, now take the soup off the stove. It has nothing to do with baking a cake. We have to remove it. Okay? The extra pieces we don't need are called introns. I know that sounds backwards. I remember introns have to leave because they are intruders to my mRNA. We don't need them. They're intruding on the protein that I want to make. The pieces that are left, well, so on my piece, I crossed out a couple of my RNA base pairs. Those are my intruders. I don't want them. So I'm going to remove them, and I will be left with just the pieces I want. Those are called the exons. They're excellent. They're what we need. Okay? This piece of mRNA is now ready to leave my nucleus and go into the cytoplasm. On Monday, we'll talk about how we read this piece of mRNA to make a protein. Pretty easy? Any questions on that? Okay, get out your notes and let's go through the PowerPoint. It's just going to take the steps I verbally told you. All right, so in protein synthesis, there's this concept called the Central Dogma. Basically, what that is fancy terms for is this is exactly what we know about DNA and RNA. And this is what's true for every organism on the planet, okay? You have a piece of DNA that generally, when you make new cells, goes through the process of replication, okay? That's what really the job of DNA is. It's to get copied to go into your other cells so that each new cell has a full copy, all right? DNA also makes RNA through the process of transcription. We know that to always be true. Every single living thing has DNA to make proteins, which means every single living thing goes through transcription first, which is what I drew on the board. In your notes after the arrows, that's where the transcription and translation go. Which translation then is taking RNA and making a protein. Okay, so let's talk a little bit about this process then. Transcription is making mRNA molecules by copying a part of DNA. This process of going from DNA to RNA is called transcription, and it's how RNA is formed. You have this picture too. Yours is not color-coded, but I would like for you to label where the green is your DNA and the purple on the inside is your new strand of mRNA. And if you all look at your picture, you will notice that this strand of mRNA is actually longer than this piece of DNA that we want to turn into mRNA. That's because this has a lot of introns. We haven't edited those intruders out yet. So here's just another picture of transcription. Here's my entire piece of DNA, and this is the gene I want to turn into a protein, so they've magnified it. The DNA strand is ATGCTAGGC. I'm going to make mRNA by adding my complementary RNA base pairs. So A to U, T to A, G to C, C to G. All right? And you have that picture in yours. So just to recap, kind of putting those base pairs together, normally when we're making DNA, we're copying it. We have A to T, C to G, G to C, T to A. In transcription, you're going from a piece of DNA to RNA. So C to G is the same, G to C is the same, T to A is the same. But if your DNA has an A, it has to pair with a U. So when we replicate DNA, what is the name of the enzyme that is in charge of laying down new DNA base pairs? Was that Christian? DNA polymerase, good. So if DNA polymerase adds new DNA base pairs, what do we think the one that lays down RNA base pairs is called? Anybody? If DNA polymerase lays down DNA, blank lays down RNA. What kind of polymerase? RNA polymerase, excellent. And remember, we have to edit our RNA. So we've made our RNA. RNA polymerase has laid down our new base pairs, A to U, T to A, C to G, G to C. We have to edit it because if you remember back to that first picture, the green and purple one, I said that that purple strand was too long. We have to get rid of our introns, our intruders. They won't code for a protein. And we leave in our exons. They stay excellent. Okay, remember, they're what we need. So in this picture down here, these are three real representations of proteins that are being made in mice, in rabbits, and in chickens. And this is just showing you the introns versus the exons. So this is an entire piece of mRNA that's been made. The purple is my intron. That's the part that we need to remove. If we remove it, we just take these two blue pieces and we smash them together. Now we have a perfect piece of mRNA to leave the nucleus and go make a protein. Okay, same thing down here. Remove these two purple, smash the three blue together, perfect piece. Okay? Lastly, and this is important to know for Monday, when I look at my final piece of mRNA, I will read it in groups of three. Those groups of three are called a codon, and they tell me what amino acid I need to hook together to make my protein. So, let's focus up here now, okay? Because I'm going to have you draw this second half in a minute, but I want to talk to you about everything. So, things we discussed. Really quickly, Brady, can you shut the door, please? Thank you. Because the bell will probably ring and you won't be able to hear. We know that every single cell in your body except for red blood cells, because red blood cells don't have a nucleus, has DNA inside of it. DNA is the code to making proteins. And the way I describe DNA is the foreign language directions to making proteins. In order for us or our bodies to understand how to make a protein, we have to take it from a language we don't get and turn it into one that we do. So we take our DNA and we transcribe it into mRNA, messenger RNA. It literally is named that because it carries the protein message to the ribosome to be made. Okay? So, in the process of transcription, you take a segment of DNA called a gene and copy it into mRNA. You can copy it over and over and over and over again, depending on how many proteins you want to make. All right? But the point is, we take our segment of DNA and we turn it into an mRNA by making the complementary strand. So if we remember, the complementary strand means that if in DNA I have an A, in RNA it will pair with a U. And if I have a T, it'll pair with an A, C to G, and G to C. Okay? So that's what I did here. I took my DNA, I made a section of mRNA. So where there was an A, I made a U. Where there was a T, an A, a G, a C, and a C, a G, and so on. Right? But I'm not done there. Because a lot of times, too much mRNA is made, and a lot of it does not code for my protein. So what I have to do is I edit it. I take out my introns. Remember, it's kind of funny that their introns get removed because introns you would think stay in. But introns are intruders. They are intruding on the DNA that I need, so we get rid of them. Everything else that I like are called my exons. They're excellent. They code for my protein. They stay in. And I compact them all together, and I get this short segment of mRNA, okay, shorter than the original, that has the ability to go into the cytoplasm where it will be made into a protein. We remember all that, right? From Friday, that was transcription. Okay. Today we're going to talk about translation. That's where we now read the directions for making a protein, and we actually turn it into one. So, my piece of mRNA travels through the cytoplasm until it gets inside of a ribosome, or rRNA. Okay, rRNA is us making it just difficult to say ribosomal RNA. Okay, so the mRNA enters the rRNA, where it is read in groups of 3. We call those groups of 3 a codon, because they code for an amino acid. All right, so rRNA reads the first codon, UGG, and says, UGG, we need tryptophan. That's the amino acid. And everybody look at the charts I gave you. Okay? Ethan, can you actually hold yours up to the screen so the people on the thing can see it? All right. So that's what our chart looks like, okay? Thank you. Here's how we read that. That is my codon decoder. All right? My first amino acid codon is UGG. So in the middle of that circle, find the big U and put your finger on it. All right, the big U, right here. My second codon is G. So within that pie piece, find the G now. No. Within the U's piece, find the G. All right, good. Okay, is everyone pointing at where I'm pointing? Look. Within the slice of the U, then find the G. All right, now we have the third G in there, or second G in there. So within that slice of the G, find the other G. And what does it say? Tryptophan, right? Good job, okay? So that's what I have up here. My rRNA goes, tryptophan, that's what we need. And my tRNAs out here, they're all hanging out around. They got different amino acids attached. The tryptophan one is going to make its way into the ribosome and drop off its amino acid. All right? Then we read the second one, CGG. So find the big C. And within that pie piece, find the G. And within that G pie piece, what do you get for the other G? Arginine. Good job. So my rRNA goes, arginine, that's what we need next. And my little tRNA right here with arginine on it makes its way into the thing, drops off its amino acid, and they form a peptide bond. This little orange bond right here, that links my 2 amino acids together. All right? Then we read our third codon, UCA. Raise your hand when you know what it is. Don't say it out loud. Just raise your hand. How many of you got serine? Is that what you're all looking at? Yeah. Awesome. Okay, so it calls out for serine. And serine comes in and drops off its amino acid. And they too form a peptide bond. And now I have a protein made of 3 amino acids that's going to break away and go do work. Whatever I needed it for. Okay? The one interesting thing that you do need to know. The last thing I need you to know is you're probably like, Miss Leone, I know that this codon codes for that amino acid, but how does tRNA know where to drop it off? tRNA has something attached to it called an anticodon. It's the complement of my codon. So if my codon here is UGG, tRNA has ACC at the bottom of it, and they know that they have to fit together. Yeah.
So it's basically like the DNA codon?
Basically, yeah. Except instead of having a T in there, you'd have a U. Okay? mRNA is what codes for my amino acid. Okay? Are we good so far? All right. Get out a sheet of paper, and I want you to draw this half of the board. Okay. So, RNA's genetic alphabet is 4 letters long, A, U, C, and G. Whereas a protein's alphabet, if you will, is 20 letters long. And so we've already seen on that chart that we were using that you go between the two of them by using the process of translation. Translating the 3 letters that make up a codon into an amino acid. So we already looked at this chart, but let's just do a little practice with it. Remember, we read this chart from the inside out. So if my amino acids are UAC, we'd start with the big U, and within that pie piece find the A, and within the pie piece for the A find the C, and find out that my amino acid is tyrosine. Let's try some more examples. CAG, what does that make? Glutamine, good. What about AAA? Lysine. What about GGA? Glycine. What about GGU? That's glycine also. What about UUC? Phenylalanine.
Yeah, that's what I said.
Good job. So, the genetic code, remember, you have a gene that you want to turn into mRNA. This is my DNA. My mRNA would be UAC, GAU, CCG. And I would read those in groups of 3, and those would code for my amino acids, okay? There are a couple special codons that you need to know. Okay, the first one is AUG. It codes for methionine. Methionine is called a start codon. Every single protein that is made in your body, maybe not in class, my examples won't always start like this, will always start with methionine, and they will always end with something called a stop codon. Okay, UAA, UAG, or UGA. During translation, the cell uses information from mRNA to produce proteins. Knowing which codon goes to each amino acid is the job of tRNA. Remember, because tRNA has the anticodon where they fit together. So, each tRNA molecule, do you give me to go back? All right. So each tRNA molecule looks really, if you were to look up at the smallest molecular view of it, it literally does look exactly like this. It's got an amino acid up top. I would like you to label that, please. And it's got an anticodon at the bottom. So if this is the anticodon, what would the codon be in the mRNA? If you look on your chart, CGC would code for what amino acid? Good. All right. So once on the ribosome, the tRNA begins binding to the appropriate site on the mRNA, so where the codon and the anticodon match up. All right. So first tRNA molecule binds down. Then the second tRNA molecule binds to the next codon, and the 2 amino acids become linked together with a peptide bond. We saw that really quickly. I'm going to pause this for a second. So it's kind of like an assembly line. This one made contact first, dropped off methionine. Second one came, dropped off proline, they peptide bonded. Now this first one can go away. That tRNA can go away. We don't need it anymore. And then it'll sort of like tick, tick over, and this one will drop down and bond the amino acids, and then this tRNA will leave, and so on and so forth until it's all done. My mRNA is going through. Here's my tRNA, where the anticodon matches the codon down here. Remember, this codon coded for the amino acid that is being dropped off, and then here is where those two amino acids are bonding together. All right, so the polypeptide chain, which is a fancy name for protein, continues to grow until one of those stop codons is reached, either UAG, UGA, or UAA. When it reaches the stop codon, the protein breaks off and is released out into the cell, where it does its job.